I'm a fisheries biologist at Pacific Northwest National Laboratory, and I'm here today to talk about the project that we've done having to do with stable isotope analyses of herring scales. So, since you all have spent some time around Prince William Sound, I probably don't need to emphasize the importance of herring in Prince William Sound. But herring are a very important resource in marine ecosystems in general and in Prince William Sound specifically. So the abundance of herring peaked in the late 1980s and early 1990s, but has been fairly low since then. So this is a graphic that depicts that. So on the x-axis here we have management year, and on the y-axis we have biomass. So you can see that herring biomass is increasing again into the late 1980s, early 1990s, when the population numbers fall quite sharply, and the population has been depressed ever since that time. So that brings about the question, what caused the crash of herring population, and what is continuing to keep the herring population at fairly low levels? And so there's a lot of different hypotheses as to why this is happening. So some things that have been floated as to what could be causing this are predation, competition, overwinter rearing conditions, fishing pressure, and disease. Another factor could also be poor foraging conditions in the fall. So herring consume a lot of food in the winter, or excuse me, in the fall, to try and get them through those thinner periods where food isn't quite as abundant in the winter, and then through the spawning season in the spring. So if there are poor foraging conditions in the fall, then that means that we might see higher rates of mortality throughout the winter. So stable isotope analysis is the tool that we're going to use to investigate some of these causes for low herring abundance. Stable isotope analysis allows ecologists to study the way that energy flows through an ecosystem. And when I say stable isotopes, that just means the isotopes of elements that aren't radioactive. That's the way in which we're using stable. So in this project, we're using carbon and nitrogen isotopes. And we're using these two elements because carbon and nitrogen follow very predictable pathways through an ecosystem. And this graphic on the left depicts that. So on the x-axis, we have the carbon isotope ratio. And we report isotope ratios in this delta notation. That's what this sign right here is. And it just means that we take the isotope ratio that we found in our sample and we're comparing it to an international standard. So that's what this delta notation means, just that we're ratioing it again to an international standard. So on the x-axis we have the carbon isotope ratio, and you can see that as we go from primary producers on the left up through herbivores, omnivores, and then carnivores, you can see that this carbon isotope ratio is increasing in a very predictable way. And the same thing with the nitrogen isotope ratio, which is here on the left. As we go through the ecosystem, as we go through the food chain, that nitrogen isotope ratio is changing in a very predictable way. So again, carbon and nitrogen isotopes allow us to follow the flow of energy through the ecosystem. So stable isotope analyses can be used to determine shifts in food webs. And this is particularly applicable with herring in Prince William Sound because we can go out and we can collect herring samples, this year or last year, and we we actually hope to compare them to those samples to the scale library that the Alaska Department of Fish and Game has. So they have a library of scales from herring going back to the early 1970s, I believe. So we're hoping to gain access to that archive to compare the stable isotopes that we find in herring currently with what some of the stable isotope values were in the past. So we're hoping to look at these large-scale shifts in food webs and how they in turn affect productivity and stock abundance in the herring population. So we have three main objectives of this project. And the first is to determine differences in carbon and nitrogen isotopes between the scales and muscles of herring collected in different seasons, and we've also collected different ages. We also want to determine the seasonal and interannual variation in the carbon of scales of individual fish. So herring scales grow by underplating. So if you think of, for instance, the rings on a tree. So herring scales are very similar in that we can see annuli. So there's one annuli formed for every year of life, very similar to rings on a tree. But when a tree grows, it's mostly adding these layers like this. It's growing outward. But herring scales are actually growing in 3 dimensions. So they're growing by underplating. So new layers are forming up like this. So the scale is growing in depth, but it's also growing in length. So we want to actually be able to look at different locations along a herring scale and see if we can see the same differences in carbon isotopes along the same scale as we can see comparing scales of different ages collected in different seasons.
I have a quick question. Is that unusual for the scales to grow under like that, or is that all fish pretty much?
That's how pretty much we hypothesize that all fish scales grow. There's a lot about scale growth and architecture that's still unknown. But that's the hypothesized method for scale growth, is by underplating. And we'd also like to determine if we can back calculate values of carbon and muscle from the measurement of carbon in scales. And again, that has to do with what we'd like to do with the Alaska Department of Fish and Game Scale Archive, looking at food web shifts over long periods of time. So for our experimental design, we looked at 4 groups of herrings. So there were fish that were collected for us by the Alaska Department of Fish and Game in 4 groups. So we had 3s and 4-year-olds collected in the spring and 2- and 3-year-olds collected in the fall. And we sampled both the muscle and scale of these herring, and we were going to do 2 different types of analyses with them. So I'll move right into the methods. So the first type of analyses that we did was with a conventional isotope ratio mass spectrometer, and that's this box here on the left. And the idea behind a mass spectrometer is that you can take a sample, put it into the mass spectrometer, and it's going to separate the atoms that are in the sample based upon their mass. So we can actually separate carbon-12 from carbon-13 to know exactly how much of a particular isotope of carbon or nitrogen is in the sample. So we take a sample of mussel, for example, and so we took the mussel fillet and we freeze-dried it and then homogenized it, and then took a very small piece of that and combusted it. So the samples need to be in gaseous form to go through the mass spectrometer. So we combusted it and then put it through the mass spectrometer. So we actually delipified the mussel, so we removed the mussel, or the lipid from the mussel by a chemical procedure. And we did this because we were comparing fish in the spring versus fish in the fall. And fish in the fall have a much higher lipid content in their mussel because they're preparing for the winter. And we didn't want that to skew the carbon and nitrogen values that we would see in the mussel. We wanted to be able to make an apples-to-apples comparison, so we removed the lipid from the mussel. We also acid treated the whole scales. So fish scales are made up of collagen, but on the top of the scale, on the outer portion, they're made up of a mineral called apatite, and that can also skew the carbon and nitrogen ratio. So we treated these scales with acid to strip away that mineral layer so that we were just looking at the collagen of the scale. We also examined just scales for carbon by laser ablation isotope ratio mass spectrometry. And I should mention that when we're doing this conventional mass spectrometry, and we're looking at the scale, we have to use the whole scale. So we can't look at discrete locations along the scale. We just have to take the whole scale and combust it and put it into the mass spectrometer. The laser ablation mass spectrometry allows us to look at discrete locations along the scale. So I know it's a bit light in this room, but hopefully you can still see these faint lines that are on the scale. So those are the annuli. So we can count those and know how old this fish is. But what we can also do is take the laser system, which is here on the right. So the laser is contained in this little box right here. And then this computer screen connects to a microscope that's inside this box. And so we can essentially point the laser at any point on the scale and sample at discrete locations. So here, on the inner part of the scale, this is the oldest material. And again, because that scale is growing by underplating, we can also see the newer material out here. So we expected to see fairly big differences in the carbon isotope ratios between the inside of the scale and the outside of the scale because those materials are of different ages. So I'll go ahead and jump right into the results then. So on the left, we have our muscle carbon-13 values. So on the y-axis, we have the carbon delta-13, and on the x-axis, we have our two groups of fish. So we have our fish collected in the fall and our fish collected in the spring. Here we have our fall 2-year-olds, our fall 3-year-olds, and then our 3-year-olds and 4-year-olds in the spring. And we were expecting to see differences in the muscle carbon values between fish of different ages. We were thinking that a fish that's 2, we would see a difference between a fish that's 2 years old, we would see a difference between a fish that's 3 and 4 and so on. But what we actually saw was the biggest differences between fall and spring fish. So these letters indicate statistical significance. So these two groups are statistically different from these two, but these two groups are not different from each other. So we saw that muscle carbon values were different between fall and spring fish. On the other hand, when we looked at the muscle nitrogen, we saw that muscle nitrogen values were different between fish of different ages. So these fall 2-year-olds, their muscle nitrogen values were different from the fall 3-year-olds. And however, the spring fish, their muscle values as far as muscle nitrogen appeared to be fairly similar. Yes?
Sorry to think about it.
Yeah, of course.
So for the nitrogen data, were those fall fish caught in different places, or were they caught in the same place? They're just different ages?
They're just different ages, but they were caught in the same place. So again, we also wanted to look at the relationship between whole scales and muscle for both carbon and nitrogen. And if we found a good relationship, we wanted to be able to apply that to back calculate values for muscle carbon using the scale archive. And we didn't see quite the relationship that we would expect. Here on the x-axis we have scale carbon, and on the y-axis we have muscle carbon. And I've broken this out into the spring on the left-hand side and the fall on the right-hand side. And you can see by these r-squared values that we don't see a very strong relationship. The relationship is fairly weak. The correlation between these between these two factors was significant, but again, the relationship is fairly weak. However, we did see a relationship that was a bit stronger in the fall as compared to the spring. However, when we look at the same data for nitrogen, so again, we're looking at scale nitrogen here on the x-axis and muscle nitrogen on the y-axis, the relationship, again, while it's weak, it's quite a bit stronger for fish in the spring and the fall when we're looking at nitrogen. So looking at the scale and muscle nitrogen seems to be very promising for back calculation. So now I'll talk a little bit about the laser ablation isotope ratio mass spectrometry. So here on the left is a picture of a scale that has been sampled using laser ablation. So each of these dots right here, each of these laser ablation marks is 50 microns wide. And each of these is a discrete sampling location. So you can see that we can really get a lot of fine scale sampling along the surface of the scale. We looked at 3 different locations on the surface of a scale. We looked at a location that was right inside the first annulus. We looked at a location that was halfway between this last annulus and the outer margin. And then we looked at the very outer margin of the scale. And so again, the oldest material in the scale is in here, and the newest material is on the outside. So we expected to see fairly large differences. But that's not what we saw. So this right here is a graph of the carbon-13 values on the outer margin of the scale. So these are our fall 2s, spring 3s, fall 3s, and then spring 4s. You can see that between those 4 ages, there's not a lot of variation. And this analysis was done on the outer margin. And we expected to see the biggest differences on the outer margin. Because if you catch a fish that's 2 and look at the outer margin of the scale, you're looking at what it was eating most recently and comparing that to a 3 or a 4. So we expected to see big differences, and we really didn't see much variation at all. But you can see by these error bars that we were seeing a lot of variation from fish to fish. And so our sample size for each group was 25 fish. So we did have a fairly large sample size. So we were quite perplexed by this result of not seeing any differences in the carbon isotope ratios on the scale, because we knew that there were structural differences within the scale. So what we did was we cut a scale in half and flipped it on the side. So we're looking at that very thin edge. And we put that into a scanning electron microscope, which is this image that we have up here on the right. So you can see that the width of the scale is, the thickness of the scale, excuse me, is 25 microns. And you can see, I hope you can see, these very faint gray lines. And so those are about 5 microns each, and they indicate those different annuli, so the different growth layers in the scale. So we couldn't see differences on the surface of the scale, but we could clearly see differences in the structure of the scale. So what we did, instead of sampling along the surface of the scale, we chose a spot at the center of the scale and used the laser to drill down through the scale to see if we could see differences in the thickness. So instead of sampling across the top, we drilled down through this 25 microns. So on the x-axis we have laser pulse, so that's the number of times that the laser hit that particular point. So this is a depth profile of the scale. And here we have, again, the scale carbon 13. So I have a spring 4-year-olds here in the black boxes, and then a fall 3 to compare it with. And so you can see that we threw out the scale, so starting from the surface and then drilling down to the very bottom of the scale, we see quite drastic differences in the carbon isotope ratio throughout the scale. So while we didn't see any differences on the surface, in drilling down through the scale, we saw drastic differences. So this means a couple different things about the scales. The first is that we thought the scales were a more static record of the conditions that the fish were being exposed to. We thought they were a fairly reliable record of the food that the fish was eating. However, it seems that these fish in times of stress or starvation are actually resorbing some of the carbon that they've stored in their scales and remodeling the scales a little bit. So these scales aren't quite the static, reliable record that we had thought. So in conclusion, we found that mussel carbon and nitrogen did differ by season and age. However, we saw little seasonal or annual variation when sampling along the surface of scales with laser ablation. But depth profiling revealed very extensive variation, and this indicates that there may be remodeling or resorption of the carbon in these fish scales. However, the correlation that we did see between mussel and scale, carbon and nitrogen, could eventually allow us to back calculate herring mussel values to examine those historical shifts in the food webs, as I mentioned earlier. And so this, I'll take questions in just a second. So this is actually a picture of the inside, a scanning electron microscope picture of the inside of a herring scale as revealed by laser ablation. So this is magnified 25,000 times. So that's what the inner collagen matrix of the scale looks like. Yes?
So your, where you drilled down, that was on the outermost part of the scale?
So that was right at the inside, because so the outermost, let's see. So the outermost part of the scale is actually very thin. Let's see if I can get, so this outermost layer, so like this right here at the top is the oldest material, and then the newer material is coming up underneath it. And so that outer material is only about 2 to 3 microns thick, so it's very thin. And what we wanted to do was we wanted to see if these scales reflected variation over the lifetime of the fish, and if we could see that. And so since we didn't see it at the 3 different locations, when we drilled down, we chose to drill through the middle of the scale, so like right about here, so that we could look at all the different growth layers. So at the very outer margin of the scale is only the most recent growth.
I'm still not clear how you could tell it was potentially resorbing then. Just because there were differences, you assume that's what happened, or?
Right, so because we saw, so when we look at a scale like this, if we sample here, you know, if we sample just on the surface like we did, if we sample here, we're in the older growth. But so this layer right here ends right about here. So we thought that if we sampled right next to where that layer ended, because the scale is growing up like this, so we have our first chunk of growth here, and then our second here, and we should be able to sample at different points and get a representation of different growth. And we thought that the carbon is being resorbed because we don't see those differences going across the surface, but we do when we drill down through the layers. So structurally, if we look at this graph, or this diagram, we're looking at a point right here on the surface, and we're looking at a point over here, and those points are part of different growth layers. So that's why we thought we would see those differences. And we don't when we just look at them on the surface, but when we drill down, there's a difference between this layer and this layer, but we don't see it on the surface. So that indicates that there's some type of surface effect, whether it's remodeling or exchange maybe with the mucous membrane that the fish has, something like that. So that's why we thought that it was resorption and remodeling of the scale. Yeah?
What would cause them to do that? Just times, I know you said times of stress and things like that, but so they need more nutrients, so they're taking it from the fat storage, and that then does their scales, or?
Exactly, yeah. So just like we saw differences in lipid content in fish from the fall versus fish from the spring, as those fish are going from the fall to the spring and they're going through, a starvation period in the winter, they're going to be, using all that fat that they've stored in their muscles, and they may actually be resorbing some carbon, nitrogen, other nutrients that they've stored in their scales as well. Yes?
Could there be a bigger difference than what you've seen by drilling through if it's not totally inaccessible, what's in the middle, from reabsorption? Because you said...
Oh, if I think I understand your question. At one point or another, each of these layers was exposed on the surface, so it could be subject to some remodeling. But as that layer is covered up, then it won't be, our thought is it won't be remodeled anymore. If that makes sense. Did I understand your question right? Any other questions? Yeah.
Can we go into the scale and muscle
Yeah.
slide? I guess I'm just trying to wrap my brain around why you would expect a relationship between scale and muscle. If they're say they're turning over, being grown at different times. I mean, muscle, they're probably turning the signature over more readily, but with scales, they're grown at certain periods. So, I don't know, I just don't know if you had something to add about that.
Right, yeah, so the muscle is going to turn over in, give or take around a month or, several weeks.
Yeah.
Whereas the scale is definitely, it's going to take a lot longer to get signature incorporated in that scale. We thought, though, since the scales are growing, they're growing every day, and they are incorporating carbon and nitrogen every day. Mm-hmm. So we did realize that there would be a lag between the carbon and the nitrogen signature in the muscle and in the scale. But we thought we could at least see that on, a seasonal time frame. Mm-hmm. And if not that, then we would still get a good relationship between ages. So even when this is broken out by age, so even if we're just looking at one age, the relationship stays about the same as when I group them together.
Yeah. It would seem like if you could get the scale signature from after a year's growth,
Mm-hmm.
like one annuli,
Yeah.
and then relate it to the muscle at that point, it might be tighter.
So that's why we were hoping that we could sample some at some discrete locations along the scale to get that kind of, that really good relationship. And, unfortunately the scale architecture didn't quite lend itself to that. So we're hoping that we can explore this scale depth profiling a little bit more and get a better technique on that to allow for that type of thing.
Mm-hmm.
Any other questions? I have some handouts that are a summary of the research as well if you'd like to take one of those. But thank you for your questions.